you for joining us today. My name is Torrin Washington, and I work in the College of Arts and Sciences Advancement and External Affairs Office. I would like to welcome you to our webinar titled The Next Wave, How Forensics Can Help Understand Virus Mutations. Before we begin, we ask everyone to mute their microphones, as this will ensure that everyone can hear our presenters. We are recording this webinar, which we plan to make available via the college's communications platforms for those who would like to watch later. If you do not want your image recorded, please feel free to disable your video camera by clicking on the video camera icon in the lower left-hand corner. Before we begin our presentation, if you have any questions throughout the talk, please write them in the chat and we will get to them during the Q&A portion of our event. Right now, I'd like to introduce our presenters. Kathleen Corrado is the Executive Director of the Forensic and National Security Sciences Institute in the College of Arts and Sciences. Dr. Corrado came to SU in January 2021. Prior to that, she spent 20 years as Director of Laboratories of the Onondaga County Center for Forensic Sciences. In addition to her service to the local community, Dr. Corrado served as the chair of the New York Crime Laboratory Advisory Committee and was appointed to 3 terms on the New York State Forensic Science Commission. Dr. Corrado earned her PhD from the University of Michigan and her bachelor's from the University of Connecticut. Joining Dr. Corrado is Michael Marciano. Dr. Marciano is a research assistant professor and the associate director for research in the Forensic and National Security Sciences Institute. Before joining SU, he was the forensic DNA analyst at Onondaga County Center for Forensic Sciences. He later joined SRC Inc., a nonprofit defense contractor company, where he focused on research and development of DNA-based applications for tagging, tracking, and locating targets. He holds a master's degree in forensic molecular biology and received his PhD in structural biology, biochemistry, and biophysics from Syracuse University. Our third presenter today is James Krill. James is Professor of Practice of Forensic Science at SU. Before joining SU, James was a senior scientist at MRI Global. He has also been an independent consultant, a research scientist at SRC, and a lab manager at Aptomatrix Inc. James is also an alumnus of Syracuse University. So this promises to be a fascinating presentation. And with that, Dr. Corrado, I'm going to turn it over to you.
Okay, thank you very much. I'm just going to share my screen. Can everyone see that? Hopefully. Someone give me a thumbs up. Thank you. All right, well, thank you all very much for being here. And I want to thank the Alumni Academy and the Dean for inviting us to give this talk. We're very excited to be here today. Okay. Just for an overview of our presentation, what we're going to do today is I'm going to give an introduction to the Forensic and National Security Sciences Institute, or what we call FNSSI, kind of tell you who we are and what we do. We'll also be giving a little bit of explanation as to how forensics relates to the study of COVID-19. We also will be talking a little bit about the COVID-19 variants and how they're tracked. And then lastly, a little bit about how forensics in general relates to pandemics. And we'll leave time at the end for questions, certainly. So where are we? Who are we? The Forensic Institute is located, our offices are located in the Life Science Building, which was built in 2009 and has state-of-the-art laboratories, both research laboratories and teaching laboratories. I think our students are really fortunate to have such wonderful facilities. We also have our faculty also teach in the, have research labs in Lyman Hall on the 4th floor. So we have a lot of research going on there as well. You just heard our introduction, so I won't talk too much about that, but in general, FNSSI has 9 full-time faculty, some of whom have appointments in other departments such as biology and chemistry. But today you're going to hear from 3 of us. My background is in forensic DNA analysis. We'll hear from Professor Krill. His background, his research interest is microbial forensics, where he studies bacteria and viruses that can be used as biological weapons. And then we'll hear, and also we'll hear from Professor Marciano, who studies bioinformatics and looks at DNA mutations and variations and how they can be used to identify human and non-human targets. We actually use a 3-prong approach to our mission of combating crime and promoting national security, and that is teaching, research, and outreach. And basically our goal in teaching is to educate and train the next generation of forensic practitioners. We also engage in cutting edge research in order to support the forensic community. And we work collaboratively with various government and private agencies in order to share information and advance the field. In our teaching, we really stress our FNSSI core values, which are science, knowledge, and integrity. And in particular, what we're trying to do with our students is to give them a very strong foundation in science and statistics. And then once they have that foundation, we couple that with a lot of very interactive hands-on courses that enable them to take that knowledge and then apply it to gain technical skills that make them very competitive for the job market and set them up for success in their careers. And thirdly, we really stress the importance of professional responsibility and integrity. In this kind of work that we do, you really can't stress enough the importance of this type of integrity and responsibility. In particular, in all of our classes, we talk about being transparent and being objective and making sure that you have that responsibility, whether it's in your work or when you testify. In terms of our academic program, we have both an undergraduate and a graduate program. And our undergraduate program is what we call an integrated learning major. And what that means is that students can't just major in forensic science alone. They have to couple it with another major. So it's like a dual major. And we actually did this intentionally. And that's because the field of forensics is just very broad and covers so many different disciplines that we couldn't possibly educate students and train them in all of those things. So basically, what this allows them to do is to get a broad understanding of forensics and see how it's applied to whatever their area of interest is, whether that's biology or chemistry or psychology. And we feel like it works out really well for our undergrads. We also have a graduate Master of Science program. And this program has 5 optional tracks. There's a forensic science track, both general and advanced. And this is a track that individuals that are hoping to go into working crime labs typically take. We also have a biomedical track, and this is a track that students use to gain additional coursework if they're wanting to perhaps pursue an MD or a PhD in the field. We have students that take the biomedical track for that. We have the medical legal death investigation track, which is basically studying the cause and manner of death. So students that are interested in pathology or medical examiner work would take that track. And lastly, we have a combined, this is somewhat new, a combined JD and MS track. And this is where students can get a law degree as well as a master's in forensic science. And this is really important because one of the criticisms that forensic scientists face is when it's used in the courtroom. In particular, attorneys and judges really don't have a background in forensic science, yet they're being asked to use the science in court every day. And so what this really does is gives these lawyers, people that are going to go out and be lawyers, a background in forensic science so that when they're using it in the courtroom, they apply it appropriately. And so we feel like that's a really important part of what we do. In terms of our students, our success of our students, we have students all over the place. We have forensic scientists that are latent print examiners, firearms examiners, crime scene investigators, DNA analysts at crime laboratories in 18 states. We also have students that are death investigators in medical examiners' offices in 7 states. We have students placed in numerous federal agencies such as the ATF, the Air Force, the Department of Defense, and the Department of Justice. And actually, if you can see in this photo here in the slide on the right, this woman is named Marie Bouget, and she is a graduate, a 2017 master's graduate, and she works for the Department of Defense, in particular for the US Army Intelligence Command. And she is stationed in Djibouti, Africa, where she basically is a firearms examiner, and she uses her firearms examiner skills to mitigate terrorist threats throughout that region. We also have a number of students that are in medical schools and graduate schools, and as we talked about previously, prosecutors and defense attorneys. In this slide, what we listed are just a few or some of our courses that we offer in the forensics program. You can see it really runs the gamut from criminal forensics to medical legal death and microbial forensics. We have, in addition to our full-time faculty, we rely on a large number of part-time faculty. And this is, again, something we do intentionally because we actually have the Center for Forensic Sciences, which is down the road. We have practicing professionals there, whether the latent print examiners or firearms examiners or DNA analysts. They work and teach in our courses, as well as pathologists and death investigators from the medical examiner's office. And this is really a benefit to our students because they're getting firsthand information. They're getting real-time case studies. They're getting, all of the technology they're being taught, we know is very current, it's state of the art. And lastly, it gives them a person that they can go to for career advice and things like that. So we really think this is one of the benefits to our programs and really makes it stand out from others. And this slide, we're basically trying to, as I said previously, point out how we have a lot of outreach in our program. We have a lot of collaborators and stakeholders that we work with, that we share research projects with, we share data with. On the left-hand side of the slide, you can see a number of federal agencies that we work with, the FBI, the Department of Homeland Security, the Department of Defense, and actually one of our current undergrads just learned that she is going to get to work at the US Army Crime Lab doing DNA analysis this summer. So that's pretty exciting. We also are partnered up with the Intelligence Community Centers for Academic Excellence. This is actually a federal program that's run by the Department of National Intelligence, and there's 13 of these centers throughout the country. And Syracuse University is one of them. And the goal of this intelligence community, the ICCAE, their goal is to increase the diversity of the pool of applicants that go into the careers into the intelligence community. And in fact, we have another undergraduate who, through the ICCAE, is going to be doing an internship this summer. However, it's at an unnamed intelligence agency. She's not even allowed to tell us where she's going to be working. We also have collaborations with a number of local and state laboratories, including the Office of the Chief Medical Examiner in New York City, the Washington, D.C. Metro Crime Laboratory, the state of Connecticut and the state of Michigan crime laboratories, as well as you can see on the right-hand part of the slide, we have a number of corporate relationships. And this is really critical. We think this offers a huge opportunity for our students in terms of being able to have opportunities to do research projects with these corporations, as well as internships. And they also help to fund a lot of our research. In terms of our research, what kind of research do we do? Well, basically, what we are looking at is we want to make sure we have the largest impact with the types of research that we study. So we're looking at the intersection of basic science, the justice system, and the national security. So here's just a list of some of the research that we have going on here in our institute. We have some trace DNA analysis, both human and non-human. We also use machine learning and artificial intelligence and couple that with forensics and DNA, latent print, and firearms and chemistry. And in fact, Professor Marciano and Professor Aderman have 3 patents that they've received in relationship to their machine learning and artificial intelligence and DNA analysis. We also study the chemical detection of synthetic drugs and drug metabolites, in particular in illicit drugs. And we also look at the biogeospatial tracking of bacteria in plants, in particular tracking the movement of drug materials and pathogens. So you might be asking, that's nice to hear about all this forensics, but how does that relate to COVID-19? Well, in reality, what forensic scientists do, forensic geneticists basically, is they exploit variations or mutations that are found in DNA or RNA sequences. And they basically study those in order to individualize whether it's humans or plants or pathogens. And so the same technology that we use in forensic DNA analysis or in microbial forensics, the same instruments, the same technology is used in identifying and tracking COVID-19 variants. And so at this point, I think I'm going to turn the talk over to Mike Marciano, and he's going to talk a little bit more about the science behind mutations and variants.
Thanks very much, Kathy. And thank you to all of you great alumni who are attending now. I'm gonna be presenting more science related to this problem set. And I wanna first say, we are not virologists, okay? We are experts in DNA, and Professor Krill will give you his background, but he is an expert in pathogen detection. So what we're going to do right now is go over the basics and how forensics gives us an eye into what we are looking at in terms of COVID variants. So hold on tight. We're going to go through a quick review of DNA. So DNA is deoxyribonucleic acid, right? It's the blueprint of life. We've heard that for ages now. And it's made of these bases, right? S, A, T, C, and G. And when it comes down to it, DNA is DNA, right? It doesn't matter if we're looking at human DNA or plant DNA or insect DNA or fish DNA or bacterial DNA. Now, of course, there are complexities to each, but when you get down to it, it's very, very similar. Now, in order to understand how these variants, COVID variants, impact the infection rates and whatnot, you have to also understand that DNA is the blueprint, but proteins or amino acids are the building blocks. So we actually go from DNA, which is the instructions for the proteins, to RNA, to the protein. So the DNA are the instructions that are in the nucleus of the cell. Then the RNA carries those instructions to help make the amino acids, which then become proteins when they're all bound together. And the shape of the protein imparts its function. So what does that all mean? So first, what are variants, right? And popular culture says, well, they're mutations. Well, variants are mutations most of the time, but it's not the X-Men, right? It's not going to give you special powers, although that would be cool. A mutation, okay, is an error that is made in the instructions that code for the protein. So right here, you see the DNA bases right at the top. You see the CAT, 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 right? And those code for the amino acids, HIS. And so this is bound together, right, and will form a protein. So what if we have a mutation, which is a variation, where the A in the DNA instructions turns to a C? So what we end up having is this trickles down, where instead of our HIS amino acid, we have now pro. So now the function, remember, we just talked about the shape imparts the function. Well, the shape of the protein will now change because the building blocks changed. So let's look at an overview of SARS-CoV-2. First, it's the official naming of this virus, this disease, was CO for corona. Okay, the VI was virus and the D disease. So SARS-CoV-2 virus was first identified in Wuhan, China. And of course, we all know because it's all over the news, and we have 131 million reported cases. And unfortunately, we have over 2.5 million deaths worldwide. So let's take a look at the anatomy. Now, I think everyone knows what this looks like by now. We really can't escape it. But what are we seeing in here? What is the anatomy of the virus? So on the exterior of the virus, we see these spike proteins, right? This is the crown that is commonly referred to. So these spike proteins are actually what we're going to be concentrating on. Then we have the lipid or fatty membrane that surrounds the RNA, which is inside of this COVID virus, this virus. So it's not a DNA virus, it's an RNA virus. And I think most of you are probably aware that 2 of the primary vaccines, the Pfizer and the Moderna, actually use mRNA as a means to impart immunity. So let's look at the spike protein a little more. So on the left-hand side, you see in red the spike protein. And this spike protein is actually what is going to help bind the COVID virus to the target cell. All right, so this becomes very, very important in the virus being able to infect and invade cells. So the mutation that we're going to primarily look at is in this spike protein, because changes in the spike protein, all right, could impart changes in how it binds to cells and how it infects. Now, one thing to note is that viruses can mutate very fast. And I am not a virologist again, but I am very certain that there are many, many, many more mutations that have occurred that end up causing this virus to not work correctly. So we're only seeing a few variants here and there that end up having an effect where they might infect more, right? Or they might not be recognized by the immune system based on the vaccines that we're getting. So if we look in, now this is as deep into the genetics as we're going to go. So this is looking at a gene, in particular, or the genome of the COVID virus. And you notice in yellow, there's something called the receptor binding domain. And this is responsible for attaching the COVID virus to the human ACE2 receptors that are on the surface of the cell. So this is the spike protein gene, all right? And there's a receptor binding motif, all right, which helps essentially create that interface. So these 2 areas where if we have mutations, right, can either cause the virus to become less infectious because it can't bind, or more infectious because it binds better, or if in fact that the spike protein is being used to impart immunity, changes in that might change an immune response. So what kind of instrumentation do we use? Honestly, we use the same things that we use in DNA analysis or nucleic acids research, robotics. So robots to help process samples at high throughput, capillary electrophoresis, which is the same instrumentation that's used in forensic labs all over the country. Single cell analyses can be done using instruments such as the DEP array or FACS instruments. And we also, and very, very important in variant tracking, is DNA sequencing, both using the traditional methods, using this, it's called the Alumni, MySeq is just one type of instrument, or a very, very cool technology that is a handheld sequencer that you can see here on the bottom right, that is the Oxford Nanopore MinION. So really interesting instruments, all of which will help us characterize and understand the COVID virus and the variants. So the CDC has approved 2 primary methods for testing, and this is antibody-based testing and nucleic acid-based testing. So the antibody-based testing, you could think of it as a pregnancy test, right, that you see commercials for. What we have is pretend these red elements here are the COVID virus, and we add this to this card, this what looks like a pregnancy test. Right? We have then binding. We have antibodies that recognize this, and these antibodies have pink dyes on them. Okay, and so what we could see is a pink line appearing on this test as these antibodies will bind this surface. So it's a simplistic way of showing how some of these rapid tests might work. And this is very similar to tests that we use in forensics every day. So the test that you have probably heard about more often is the PCR based test. And you can see on the left side of the screen there, that's a PCR instrument. There's really not much to it. It is a heat block that changes temperatures from high to low. And what we could do is we could take a small amount of starting DNA or RNA and start to create more identical copies, So it's very sensitive, meaning we could detect low levels of COVID using this PCR test. And that's why it's one of the gold standards, because false negatives, right, are very uncommon in this. So let's look at the variants of concern. So the UK variant is fast becoming one of the predominant strains in the world. It's 50% more transmissible, and vaccines, though, have been noted as still effective. There's the South African variant, the Brazilian variant, and this Californian variant. So a lot of these have these mutations in the spike protein. And if we look a little bit more closer at how these variants are penetrating the global population, we see from the beginning in January on the left side, right, to April, the UK variant, the purple, is representing a huge percentage, almost 50% of all cases. Okay, the California variant, similarly, is steadily growing in terms of prevalence. So it's important to remember that these variants, right, these variants are present, and DNA sequencing, which is the main tool to identify these variants, is occurring and actively being monitored. And so I'm going to turn it over to Professor Krill right now, who's going to give you some really interesting background on microbial forensics.
Hi, everybody, and thank you, Dr. Marciano. Let me just pull up my screen. Great. Can everybody see this? Give me a thumbs up. Awesome. Great. Okay, so we'll start off on microbial forensics, and I'll give you a little bit of definition, maybe a little bit of historical perspective, and then we'll get into really what the infrastructure is like for microbial forensics. And please keep in mind, microbial forensics is a fairly new field of study, really kicked off in 2001, and we'll talk about that in a little bit. So Dr. Bruce Bedoli, the godfather of microbial forensics, defined it as having a scientific discipline dedicated to analyzing evidence from bioterrorism act, biocrime, or inadvertent microorganism toxin release for attribution purposes. And attribution really being a key term here, meaning where did it come from? The main areas of interest for microbial forensics starts out at biosecurity, which is an area that historically I've worked in looking for biological agents that could be used as weapons. There's public health forensics. We also track foodborne outbreaks, source tracking for wastewater, human identification using the microbiome, and also estimating the post-mortem interval using the microbiome. So a very broad spectrum. And speaking of spectrums, one of the things that we really try to do is differentiate between natural outbreaks and things like accidental releases, biocrimes, bioterrorism, and biowarfare. So if you look at the spectrum of biorisks, you might be able to think a natural outbreak could be potentially confused as an accidental release, and so on and so forth. And an accidental release could be considered, maybe that was a biocrime, maybe it was an act of biological warfare. And as we move down the spectrum of biorisks, things get a little bit more scary. Historically, the first real act of biological warfare, documented anyways, was in Crimea 1346 under the rule of Mongolian Golden Horde. Tartar forces were catapulting plague-infected corpses over castle walls at Kaffa. You see this terrifying catapult here on the left. So there's a lot of speculation that this is where the Black Death actually started in Europe. We can see here from 1346 to 1351, the spread starting from Kaffa, and this is over several years. So by today's standards, this spread would be considered pretty slow. So what happened? Well, globalization happened, right? This follows traditional trade routes in the 1300s, but now that we have airplanes, global travel, many more trade routes over the sea and through the air, spread becomes much more rapid. And the Black Death is widely considered to be the greatest public health disaster in recorded history. And to give you a little bit of perspective, the world population went from 475 million to about 375 million. So that's about 100 million people died during that time. And now let's take a leap into influenza. So influenza can cause a lot of deaths. It creates a high economic burden, but hasn't been known to be used as a biological weapon. So if it's not a biological weapon, why would microbial forensic scientists be interested in influenza? So any research that involves an alteration of the pathogen that results in a phenotype that aims or is expected to increase transmissibility or virulence is considered gain-of-function research. And this is done generally to understand transmissibility, understand the emergence, forecast reemergence, virulence, and understand host interactions. So can you think of why this might pose as a security risk to the United States? And I generally ask people to, for a moment, put your bad guy cap on and really think about it. It's a nice place to visit, or an interesting place to visit, but not a place you want to live, right? So while using these techniques can improve our understanding of the influenza virus, gain-of-function research poses a biosecurity risk because of its dual-use nature, the ability to have both beneficial and malevolent purposes. And an example of this is the reconstruction of the 1918 influenza virus. So the full genetic sequence has been determined, but reconstruction of the virus is actually necessary in order to determine its phenotypic characteristics. So researchers have done this, and consider, should the results be published? Should we put out a blueprint that tells people how to do this kind of work? And I know my personal feelings on this, but the research was published. And now you might be asking yourself, why am I bringing up 9/11 during this time? Well, this is really where microbial forensics got its start. Originally, there were concerns of a biological weapon on board one of the airliners. So the FBI responds to all 3 9/11 crime scenes. The CDC starts monitoring local hospitals. The Department of Energy deploys an aerosol sentry at all the sites. All testing was conducted at USAMRIID, which is the U.S. Army Medical Research of Infectious Disease. It's located in Maryland. And samples were collected hourly. So there were no biological weapons on board any of the airplanes. But a week after 9/11, you might recall Amerithrax, where anthrax letters were sent to various news stations and government office buildings, and eventually infecting several people and killing a handful. So we actually had a bit of infrastructure in place because of 9/11 to start dealing with the Amerithrax case that happened a week later. So the FBI had formed the Technical Hazardous Response Unit, which is by design composed of personnel that have expertise in health and safety, public safety, and military. They provide evidence access and actual logistical support for FBI investigators and other folks in the CDC. The CDC had still has a large role, and I know you probably hear about the CDC and the decisions they're making about what we can and can't do as far as handling the virus and so on. One of the agencies that they have is the Bioterrorism Preparedness and Response Program. So they developed a strategic plan and guidelines for safety equipment, standard laboratory practices, and a lot more. And a lot of these practices are carried over and still practices that we use today, even when dealing with COVID. And of particular interest is the Laboratory Response Network. They have been very responsive to the COVID outbreak. So it's a complete laboratory network throughout the United States and the world, really, that is responsible for responding to bioterrorism events, emerging infectious diseases, chemical terrorism, and other public health emergencies. It gives them a very broad spectrum of accountability. They're composed of federal, state, and public health labs, military labs. They deal a lot with food testing, environmental, veterinary, and international. And what hasn't made the list here is most recently they've been accepting academic labs as part of the laboratory response network. So laboratories like ours here at Syracuse University could actually be considered to do some of this research in really tracking down variants, as long as we have the capability and the capacity. So I think that loops everything together. And I'd like to give a special thanks to the Alumni Academy, Elizabeth Cole and Dr. Roulant, and open the floor for any questions. Thanks again.
Well, thank you all. That was incredibly interesting and informative. And so, yes, I would like to open it up for questions now. So if you have a question for our speakers, please put it in the chat box. If you have a question for a particular speaker, please feel free to include their name and I'll make sure to direct it to them. And so looking at the chat, the first question that had come in, I think it might be for you, Mike, is do you use CRISPR?
So in terms of the research, no, but actually that would be a question I'm going to pass over to Professor Krill.
So I also don't use CRISPR. However, I'm very interested in being able to identify organisms that have been modified by CRISPR. So it's something we're looking into. And there is a bit of tracking that we can do in the genetic sequence, a lot of these techniques like CRISPR don't introduce a lot of scarring, but we might be able to look at things like methylation and stuff like that of the nucleic acids, be able to tell if something's been genetically modified. There's nothing more terrifying than a genetically modified microbe that's able to infect more or higher virulence and so on.
Thank you both for that answer. I have a couple questions that came in separately from this, and so this can be addressed to any of you. So what roles do you see forensics playing as the pandemic continues, and does that role shift from beginning to middle to end?
So I have to say one interesting thing that was brought up by our crime scene professor, Professor Adrian Grenier, who actually worked for the state police for, I think, 27 years as a crime scene investigator. He came to me and said, and this is sort of a component of the pandemic, right? Not directly related, it's indirect, but with people wearing masks all the time, is it more or less likely that we'll find DNA evidence at crime scenes? A really interesting question if you think about it, right? Well, if they take their masks with them, right, we might not. But if they leave their masks there, right, the saliva we could collect potentially. But it's just an interesting side note to this pandemic, how some of the indirect effects can affect our daily life as forensic scientists. That's just one small aspect I wanted to bring up.
Yeah, and I'd like to add to that, if that's okay. If we think back to the spectrum of biorisk that I showed you at the beginning of the pandemic, there was a lot of interest in determining, was this potentially an accidental release from a lab versus a natural occurrence. And to date, there's no data, at least not publicly available, that suggests that it was an accidental release. But that's something microbial forensic scientists are very interested in being able to determine. I think it's something that my lab really likes to focus on, and the research is getting better.
Well, thanks for that. We did hit a question in the chat. Can we tell from the structure of the COVID-19 virus if it was man-made? So
that's going to be one of those things where ultimately we're going to need to know the DNA sequence or the RNA sequence and compare that to others. As Professor Krill was saying, there are potential methods of determining if something was man-made, but first you have to eliminate the possibility that it was a variant or a mutation that occurred. So I'm not saying that you cannot. I'm just saying that based on my experience as a DNA analyst, you'd have to rule out first that it was a natural phenomenon.
Gotcha. And just got another question privately. Are the antibodies produced by vaccines different from the antibodies produced by an infected person? If so, is there an antibody test for these to make sure the vaccination shot was effective?
Yeah, I mean, I can maybe answer that. So when we manufacture antibodies, we're basically making what we call polyclonal antibodies, that we put the protein in, and the antibodies are actually recognizing all different areas of the protein. And so while they're different than the antibodies that your body manufactures, they're both being made targeting the same protein. It's just that they might target different areas of the protein. So they are different, but the tests that we're using right now are basically, the antibody test is basically just testing, do you have the antibodies inside your body? So if you got the vaccine, you likely will have antibodies that will react.
Thanks for that. Another question in the chat. So with existing COVID variants, can you extrapolate possible upcoming variants or likely ranges? And how well can you prepare?
Jamie, go ahead.
So that's a very hard question. There's a lot of research going in right now using machine learning, and maybe Dr. Marciano can speak to this, but to really kind of understand and project what the worst case scenario would be for variants and genetic mutations. How can we prepare? Well, you know, being able to administer rapidly and develop rapidly new booster shots and so on would probably be one of the best things we could do.
Let me just add that there are things, there are areas of the DNA, RNA, that are mutational hotspots based on a variety of reasons. So, you know, I can't talk specifically in COVID, but there are areas of our DNA even that are more prone to mutation for a variety of reasons. So it might be possible to extrapolate, but again, not a virologist.
I like that disclaimer there. You're not a virologist. I love it. So, so I got one last question, and then I think we can wrap it up. So, so does the role of forensics in the tracking of COVID or other viruses vary greatly from the U.S. versus other countries? And where do we fall in that spectrum, and how we utilize forensics training and knowledge compared to other countries' prevention and response efforts? It's a tough one.
It's a great question.
I can give an example, I guess. So I was deployed overseas with the Department of Defense in a mobile laboratory, and one of the first of its kind, trying to detect microbial viruses and bacteria that could be used as potential weapons. And as far as the technology goes and what we're able to do, we're pretty advanced. And I don't want to hazard the guess saying we're more advanced than some of the other countries. I was able to work with French forces and British forces, and even Hungarian forces that had advanced technology as well, and we really put our heads together. I think the beauty, well, it's, you know, hard to say the beauty, but the beauty of something like this can be found in the fact that we can work together and combine all of our efforts and really drive something forward when we need to. I don't know if that answers the whole question.
Good. No, I appreciate that. So thanks. So I just had one last question, and this is more of a Monday morning quarterback type of question, but what would you have done different if you were in charge of the pandemic response? And this is for any of you if you want to answer, without getting too political.
I think I'll just say one thing for sure is that I think that there was a little bit of a delay in tracking, paying attention to the new variants coming. I think everyone was just so focused on, you know, what can we do to stop the spread right away and everything. So it just, I think we were a little unprepared at first, and that was our first defense. And then people started to realize, wait a minute, we should have been sequencing these variants from day one. And so there's a little bit of a delay. So I think, you know, if we had seen that, we would have seen things coming a little bit sooner. But, you know, again, that is a little bit Monday morning quarterback, because I don't think we really were expecting this to happen as quickly as it did.
I agree completely.
I agree as well. I think, you know, global sharing of information, although very sensitive and challenging. I think if we were sharing information, if other countries were sharing information as well, a little bit more openly, the response might have been a little bit faster.
Yep. And let me just end this on just perspective on what Dr. Corrado said. You know, with the variants and with the vaccinations, and this is not a comment, you know, pro or against. It's just a fact. In that, what you could look at us as is as petri dishes, right? And if we let the virus, if we're infected with the virus, it gives the virus a chance to mutate and create these variants, right? So the more people that are immune to the virus, the less chance these variants can even develop. So I think that one of the issues is people not understanding the need for immunity and what immunity can impart to the defense of, I guess, the human race against COVID going forward. And it becomes very confusing because there are a lot of different messages from the CDC, from the White House, from, you know, your county health departments. So communication, I think, is also the key.
Well, thanks for that. We actually got one more question that came in the chat. So, are the Chinese and Russian vaccines more traditional approaches or RNA based?
I have no idea. I'm
Let's just
sorry.
say that. Do you guys know that? Yeah.
I don't know either. I will say that these RNA-based vaccines are really incredible, and I think we're going to be seeing more of that. I mean, they're now looking at them to be used for cancer treatment and things like that. So I think this is the wave of the future, and that's all we're going to see moving forward.
To be continued, right? So, well, with that, I'd like to thank our fantastic speakers. So thank you for putting together this presentation today and sharing your expertise with us. I also want to thank you all, our guests, for attending this afternoon for our final Alumni Academy of the semester. We really appreciate you being here. And we will be hosting more of these as we move closer to the fall semester. So stay tuned for more invitations from the college. Thank you again for joining us. We hope you enjoyed this event and we hope to see you again soon. So have a great night, everyone.